any diet that is going to get people to eat less processed foods and more filling, satiating foods is going to be a diet that does better than the standard American diet. So I have no doubt that there are people who go on carnivore and they get healthier. The question really becomes, why did you need to do that? Could you do that and still have some dietary fiber and get the benefits of that? I have had carnists push back against me and say, well, dietary fiber is just filler.
Fiber is found in every single plant on the planet. By definition, when we go carnivore, we're not eating plants anymore. And why does he think that fiber is so magical anyway? It's not like we should just try and extract fiber out in isolate and have that with our steak. Now it's no longer a whole food, putting something in the body that's indigestible, sugars we can't digest. So I don't understand why he can't get away from the fiber fallacy. And it's just something that ends up down the toilet. Yes, there is a benefit once it hits the colon, but we get these same benefits in a more thorough way without the inflammation, without blocking nutrients, without fermenting this stuff and causing all this gas, hydrogen, carbon dioxide and methane. So yeah, if you want to eat fiber, just go outside. It's absolutely everywhere.
Yummy. What does fiber do? It produces short chain fatty acids, and that's a saturated fat. This is him all over. Complete and utter contradiction, because in one tweet he's saying saturated fat. It's got to be bad for you, right? But why don't we eat some fiber and that'll make you some saturated fat? So what is it? Is it good to have saturated fat, or is it bad to have saturated fat?
I don't even absorb it. In fact, in carnivore, I don't even poop as much because, I'm absorbing so many more nutrients and blah, 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 blah.
I don't like the way you're looking at me right now.
And I always find these really Olympic level mental gymnastics interesting, because it's not.
You see, now, this, what I take issue with is the clinical experience of real people, blah, 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 is how he looks at it. And that's him all over. Does it not actually matter to him that people are saying these things, because that's what they're actually noticing? They're getting away from the fiber, and all of that stuff is true. We are absorbing more nutrients. We are pooping less. We are having less GI distress, and they just write it off with things like blah, blah, blah.
Not like we don't just have tons of studies in humans looking at this with actual hard human health outcomes.
Okay.
So is the primary concern that you're worried about from carnivore low dietary fiber?
That's one. And then a lot of them are choosing very fatty cuts of meat that are high in saturated fat. And there is a big debate amongst diet tribes about whether saturated fat is bad for you, because it raises LDL cholesterol. And there's a debate around LDL cholesterol. And people,
you see, you've gone into the realm of proxy markers. This is the other thing as well. A causes C, when in fact A might cause B, then B causes C, and they just link it all up. It's incredible that saturated fat and LDL comes into this as negatives.
Yeah, and it's like what you've just said. He's just debunked himself. He's gone from saying fiber's good and saturated fat's bad. Let's talk in calorie terms, because he loves that word. So if we're talking about what sort of apes and ruminants get 70 to 80% of their calories from, it's saturated fats. They turn the leaves and the grass into saturated fats, because that's what it does when you can actually accommodate that stuff in the body and metabolize it properly, ferment it thoroughly. That's what happens. That's what you derive. And then, a lot of people on carnivore eat fatty cuts of meat because we don't want to die of rabbit starvation, number one. But number two, you wouldn't go out in the wild, find a nice beast and cut all of the fat off. That makes no sense whatsoever. The fats and the proteins are metabolized best when they come in the body at the same time, like an egg. It looks like 50-50, the egg, white and the yolk. You get absolutely everything you need. It's more anabolic as well. So it's just crazy. He's debunking himself in real time.
well, carnivore will say, well, I feel great. You can feel great right up until you have a heart attack.
Oh, shiver my timbers.
So looking at LDL cholesterol, what really changed my mind on it, because again, I was in that camp, was what were called the Mendelian randomization trials that started coming out around 2008, 2009. Now, Mendelian randomization is where they look at your body. People's bodies naturally have like polymorphisms on genes. So you'll have people who secrete naturally more or less LDL.
So he's going to talk about Mendelian randomization as if it's the be all and end all and proves cause and effect. But of course it doesn't. It gets absolutely riddled with holes. For the first thing, let's get it basic for everybody. A SNP, a single nucleotide polymorphism, which is just DNA is just changed somewhat, or mutated is the word they like to use. What they try to do is find an association. That's the first thing they do. So they're going to say, right, this SNP, as they call it, is it associated with anything? So we're straight back to epidemiology, and then they will assume that they found some sort of connection. And then they will assume and do what I just said about proxy markers, that if this person has this SNP, therefore they're going to have high LDL, low density lipoprotein, which contains cholesterol. Then they're going to find another group that doesn't have this SNP, and they're going to assume that they have low. And a lot of times, actually, the cholesterol or the LDL is not even measured. There's a lot of assumptions going on. And then you've got the problem with hidden genetic connections or gene connections. Because if you're studying genes, you could be looking at a gene of interest, and a nearby strand, another DNA strand or some genes later on, might actually be the thing that's causing this variation. They don't know enough about the human genome to be able to say, yeah, it's definitely this. It's a bit like, if there's 10 kids in the class and you, Lee, are always making lots of noise, and then one day I make a really big noise and smash a window, you get the blame because you're associated.
Areas of metabolism. So what you have essentially is a great lifelong randomized control trial of people exposed to more LDL cholesterol versus people exposed to less LDL cholesterol.
Yeah, but you don't isolate everything else. It's not a group where there's a bunch of identical twins with the same genetics and the other twin of that person who don't have the SNP. It's not that, is it? So immediately you've got confounders, you've got covariants, you've got collinearities. It's just rubbish. It's not science.
And when you look at that, the exposure to lifetime LDL cholesterol, you can basically draw a straight line through it between
lifetime exposure to LDL cholesterol. So that is assuming then that they did these blood tests every single day. So from birth, every single day that we got their lifetime exposure to high LDL. No, we don't know that. And then when you talk about this straight line, whenever you look at the straight line that they produce, it's full of adjustments. You look at the graphs, there is, confidence intervals as long as your arm. So yeah, we've got this straight line, but actually there's something above, there's something below, there's something above, something below. And when you look at what's called the R squared value, which is when you look at sort of the area under the curve and you do some calculations, the significance is really poor because of those confidence intervals. The chart looks compelling unless you know what you're looking at. And then you realize, oh, hang on a minute, it's all smoke and mirrors.
And the incidence of heart disease. Now, some people have said, well, it's not LDL. You don't have to worry about the large LDL. You got to worry about the small oxidized LDL. I don't know if you've ever heard this argument. It's a very popular argument. What is true is that small oxidized LDL can more easily penetrate the endothelium, but it deposits less total cholesterol. Large LDL does not penetrate it as easily, but it still penetrates it, but it deposits more cholesterol per LDL particle. The net effect is both are equally atherogenic.
Now we've got to ask, why is it trying to get into that space? When he said the small one's getting in and the large one can get in, and because it's bigger, has more cholesterol in it. Well, why is that happening in the first place? There's damage to the endothelium. There's damage to the interior of the artery. Now, we can disprove all of this by looking at doing a thought experiment and thinking, well, where do all the plaques form? Well, they always form in arteries, but they don't form in veins. Well, why is that? It's the same LDL particle going around. It's the same cholesterol within that particle, but there's no plaques in the veins, but they're definitely in the arteries. Well, not only that, they're not all over the arteries. They're in particular places of the arteries, and they're predictable places. So one effect is not actually having the cholesterol in there. It's what's causing the damage to the wall of the artery. And normally that is some sort of injury because of inflammation, because of high pressure, because of the turbulence of the blood, because of what we're eating, which is inflammatory. Well, the pressure in an artery, everyone knows about 120/80, and the vein, it's about 5 to 10. So the pressure is like 1/10. That's the first thing. The blood is flowing in a more laminar form. It's much easier to get blood out of an artery. So we could look at the vascular tree and say, right, that's where the plaques are going to form. So it can't be the cholesterol, because if it was the cholesterol, if it was LDL, it would form in the veins as well. Or it can't be the cholesterol because actually, even in the arteries, it doesn't form plaques everywhere. It's in certain places. But what it could be is injury to the interior of the artery, and cholesterol is there. Is it a bystander? Is it there to repair? And then the common sense as well, when you look at people with hard outcomes, which you can't fudge, that's people actually dying. Over 50% of people who die of heart disease have normal or allegedly low cholesterol.
In your experience, when people go carnivore, what happens to their blood pressure?
Well, it actually lowers. If you just ran up the stairs, I would hope your blood pressure would go up because your body would demand more oxygen. we also need to realise that these arbitrary numbers of 120/80 aren't for everybody. I mean, why would a 12-year-old girl have 120/80 and an 80-year-old man who's like 200 pounds? We want to actually be more realistic with our numbers, and that's another one where I think the 120/80 is too arbitrary. I think it's based on a 21-year-old male athlete. But anyway, when they go carnivore, the blood pressure normalizes. I think that's the better thing to say.
You're right.
You don't see
right.
lots of, highs and lows. And, if you're taking part in sport, then it will go up appropriately. And if you're resting, then it will go down appropriately. But we don't want chronically elevated high levels of blood pressure. But on carnivore, never seem to see that problem. In fact, normally within about 3 weeks, people will say, I took your advice. I kept monitoring it. I spoke to my doctor, and we're looking at either reducing the medications or coming off the medications, because that is one of the things with carnivore. You have to say to people, you're on medications, your disease is going to reverse. So you need to speak to your doctor, your healthcare provider, whoever it is, keep them in the loop, because you want to make sure that your dosage is coming down and down and down.